Now it is visible.
Yes, ma'am.
So we have seen the structure of ribose sugar, and then you need to know also the nitrogen bases structure, molecular structure. So what happened that there is that nitrogen basis pyrimidine is there and purine is there. And purine bases, there are two type of purine bases, which is adenine and guanine. And there are three pyrimidine bases, which is thymine, cytosine, uracil. Hmm. And purine, what is the molecular structure of purine? It is made up of 6-member ring fused to a 5-member ring. You can see that 6-member ring and it is fused with the 5-member ring. Okay, 1, 2, 3, 4, 5. And the 6-member ring, that is 1, 2, 3, 4, 5, 6. And pyrimidine, it is only consists one ring which is made up of 6 carbon. And the rings are not only made up of carbon, specific formulas and structure are not required for IB. So it is not only made up of carbon, they consist nitrogen also. The ring in the ring structure only carbon is not there, nitrogen is also there. So you can see that this is a polypeptide chain of RNA and polypeptide chain of DNA. And you can see that in DNA base is there, and it is connected with the deoxyribose sugar, and this deoxyribose sugar is connected with the this phosphoric acid. When the deoxyribose sugar at its 5' carbon atom, it is joined with the phosphoric acid, then that end is the 5' end. And when the 3' end of the sugar is joined with the phosphoric acid of previous nucleotide, then the end is 3' end. So polynucleotide का 5' end क्या होता है और 3' end क्या होता है? अगर हमारा जो शुगर है वो 5' एंड कार्बन इसका कार्बन जो है फॉस्फोरिक एसिड के साथ कनेक्ट होता है, दैट एंड इज द 5' एंड. एंड द 3' एंड ऑफ दैट शुगर इज कनेक्टेड विद द फॉस्फोरिक एसिड, दैट इज द 3' एंड. So this is the DNA structure and RNA structure. The difference is the sugar mainly, and ribose in the RNA and deoxyribose in the DNA. एंड नाइट्रोजन बेसिस के pyrimidine में भी डिफरेंस होता है आरएनए और डीएनए में. तो दिस इज द मॉलिक्यूलर स्ट्रक्चर जिसकी मैं बात करी थी, जो आपको पता होना चाहिए क्या होता है मॉलिक्यूलर स्ट्रक्चर. द होल स्ट्रक्चर यू शुड नो. We will start from adenine. So you can see that this 6-member ring, it is made up of carbon, nitrogen, carbon, nitrogen, carbon, carbon. And this 5-member ring is there. So where should be the double bond in the adenine 6-carbon ring structure, and where should be the double bond in the 5-member ring in the adenine, you should know. And what is the side group in the adenine, which is NS2 group. NS2 group at the 1 carbon atom. And here you can see the guanine, there is 2 side group is there, that is NS2 and C double bond O. So this is the guanine structure and adenine structure, which is purines, and there is the uracil, cytosine, thymine. So these pyrimidine consist the 5-membered ring, and they also consist the side group. In cytosine, there is a amino group and keto group is there. In uracil, there is 2 keto group is there. In thymine, there is 2 keto group plus 1 methyl group is there. So you should know the structure, the molecular structure of purine and pyrimidine. So, and how the structure is numbered. You can see that in purine 6-membered ring and 5-membered ring, and it is numbered from this nitrogen 1, 2, 3, 4, 5, 6, and then 7. 7 नंबर किसको दिया जाता है? 5 मेंबर जो रिंग होते हैं उसके नाइट्रोजन को, टॉप मोस्ट नाइट्रोजन को नंबर दिया जाता है 7, then 8, then 9. Similarly, in adenine, at the 6 carbon atom, there is amino group is there. In guanine, at the 6 carbon atom, there is keto group and amino group is there. And here in the pyrimidine, how the number is given? This nitrogen, the lowermost nitrogen is given as 1 number. So 1, 2, 3, 4, 5, 6. And in cytosine, at the second carbon and 4 carbon, there is keto group and amino group. And uracil, at the second and 4 carbon, both carbon consist keto group. And in thymine, the 2 keto group is there, similar to the uracil, but 1 methyl group is there at the 5th carbon. So this is the whole structure. So I'm not going in the tautomerism. So what is the function of DNA? So nucleotides are precursor of the nucleic acid, DNA and RNA, and nucleic acid are concerned with the storage and transfer of genetic information. So all of the genetic information, all of the hereditary information is stored and it is transferred to another daughter cell or another the progeny, and that genetic information is present inside the DNA. And apart from the nucleotide, which is the part of the DNA and RNA, nucleotide is also the part of currency of energy. ठीक है, एनर्जी करेंसी का भी पार्ट होता है न्यूक्लियोटाइड, लाइक एटीपी which is currency of energy, and it is made up of adenosine triphosphate. So it is termed as it is ATP and it is made up of nucleotide. So nucleotide are the part of the currency of energy. Nucleotide are also the part of coenzyme like NAD, FAD. ठीक है, NAD क्या है? निकोटिनामाइड एडेनिन डाइन्यूक्लियोटाइड. FAD क्या है? फ्लेविन एडेनिन डाइन्यूक्लियोटाइड. So the nucleotide is the part of currency of energy. Nucleotide is also the part of coenzyme NAD, FAD, and some metabolic regulators such as CAMP and CGMP. So A stand for adenine mainly, G stand for guanine, C cyclic. MP monophosphate, here also monophosphate. So there are coding and non-coding region found on DNA. Now again we come back to the DNA. So DNA जो है, DNA जेनेटिक इंफॉर्मेशन कैरी करता है, DNA न्यूक्लियोटाइड से मिला के बना होता है, और DNA में कोडिंग और नॉन कोडिंग रीजन्स होते हैं. कोडिंग रीजन्स वो होते हैं जो आगे जेनेटिक इंफॉर्मेशन को ट्रांसफर करते हैं डॉटर सेल्स में. So coding region code for genes or proteins. Non-coding regions can be either DNA junk or help regulate protein synthesis. ठीक है, तो डीएनए में हमारा कोडिंग रीजन होता है, नॉन कोडिंग रीजन होता है. कोडिंग रीजन मेनली जो है ट्रांसक्राइब होता है, ट्रांसलेट होता है प्रोटीन फॉर्म में, और नॉन कोडिंग रीजन जो है इस ट्रांसक्रिप्शन और ट्रांसलेशन में हेल्प करता है एज अ रेगुलेटरी प्रोटीन, और या तो ये डीएनए जंक की तरह रहता है, होता है. So Watson and Crick proposed in 1953 the three-dimensional model of DNA structure, and that accounted for both the X-ray data and the characteristic base pairing in DNA. So X-ray data के साथ मैच किया था ये DNA structure जो इन्होंने propose किया Watson and Crick ने. और what is the main point in the Watson-Crick model, DNA double helical model? It consists two helical polynucleotide chain we already discussed, two polynucleotide chain coil around the same axis to form a right-handed double helix. So these two polynucleotide chain coil around the same axis and they form the right-handed double helix. And in this helix, the two chain or strand are anti-parallel, means have an opposite polarity. So you can see this is the right-handed double helical structure, and in this right-handed structure, the two chain are anti-parallel. What is the meaning of the anti-parallel? It is the meaning that the one helical chain that is in the 5' to 3' direction, and the another chain is 3' to 5' direction. So they bear the opposite polarity. And that characteristic is termed as the anti-parallel structure of DNA double helix model. And backbone of each chain, which consists of alternate sugar phosphate residues, and they are outside of the double helix facing the surrounding. So the backbone consists the base plus the phosphate plus sugar, and they face the surroundings. And the purine and pyrimidine base of each strand face inward. तो आउटवर्ड क्या होता है? आउटवर्ड होता है जो कि सराउंडिंग को फेस करता है, वो होता है आपका शुगर एंड फॉस्फेट, और जो इनवर्ड मॉलिक्यूल होते हैं वो क्या होते हैं? वो प्यूरिन एंड pyrimidine बेसिस होते हैं. So these yellow color, you can see these are bases, and this is phosphate, which is linked with the this yellow is the sugar, this is phosphate, and sugar is connected with the nitrogen base, and the two chain are connected with each other by the nitrogen bases. And the base pair are 0.34 nanometer apart in double helix. A complete turn of helix takes 3.4 nm. So one complete turn from this to this, the distance between the one complete turn is 3.4 nanometer. And in one turn in the bDNA, there are 10 bases which are present. And 10 बेसिस के बीच जो डिस्टेंस है वो 3.4 नैनोमीटर है, तो दो बेसिस के बीच जो डिफरेंस होगा दैट obviously that is 0.34 nanometer. So the helix, and the another property is helix has two external groups, that is major groove and this is minor groove. So the narrow groove is called as minor groove, while the wide groove is called as the major groove. The major groove is the site for DNA binding protein. So DNA binding protein can bind to this major. So you can see the overall structure of the DNA. It consists double helical chain and it is right-handed helix, and one turn of the helix distance is 3.4 nm, and in one turn of the helix there are 10 bases, and the distance between the two bases is 0.34 nm and nanometer. And there is a minor group is there and a major group is there, and in the major group the DNA binding protein, they are attached. So the pairs of bases are always between a purine and pyrimidine. तो जो पेयरिंग होती है बेसिस की वो प्यूरिन और pyrimidine के बीच होती है. A जो है T के साथ बाइंड होता है, G C के साथ बाइंड होता है. And this is the rule according to the Chargaff, who has given this rule. And there is a hydrogen bonding which occurred between the bases. 2 hydrogen bonding between A and T, and 3 hydrogen bonding between G and C. And two chains do not have the same base composition. The two chain are complementary to each other. Such chain are called as complementary chain. So complementary क्यों होता है दोनों chain एक दूसरे का? क्योंकि A T के साथ bind होता है, G C के साथ bind होता है. These are called as a complementary chain. So for today, this is enough, and then in the next class I will tell you about the types of DNA, A, B, Z DNA. उनमें क्या differences होते हैं. आज तुमने DNA structure and function देखा, तो please go through the book for studying this DNA structure and function, and please study the type of DNA also. And in the next class I will start with the questions from the DNA structure and function, then I will tell you about the type of DNA. Meaning your attendance. Tanishq,
Present, ma'am.
Preeti,
Present, ma'am.
Sanya,
Present, ma'am.
Riya,
Present, ma'am.
Raghav,
Present, ma'am.
Rajan, Pratiksha,
Present, ma'am.
Riyaman, Kinshuk,
Present, ma'am.
Roshan,
Present, ma'am.
Madhur,
Present, ma'am.
Hansa, Simran,
Present, ma'am.
Neeraj,
Yes, ma'am.
Priyanka,
Present, ma'am.
Tuhin,
Present, ma'am.
Falesh,
Present, ma'am.
Mohit, Nitesh, आशीष, सारे गायब, प्रदीप, अभी इसके पहले 16 बच्चे थे, नाउ 14 स्टूडेंट्स आर हियर। और जो भी ज्वाइन नहीं करता है, आगे के टेस्ट में भी मार्किंग उसी हिसाब से होगी। अटेंडेंस इज मैंडेटरी, ठीक है। सारे टॉपिक्स हम कवर करा रहे हैं, और टॉपिक्स के कवर करने के लिए आप लोग को क्लास ज्वाइन करना पड़ेगा। और बीच बीच में मैं सरप्राइज टेस्ट भी ले सकती हूँ। इंटरनल टेस्ट तो होंगे ही होंगे। इंटरनल टेस्ट में आपके अटेंडेंस का भी ध्यान दिया जाएगा, ठीक है। चलो, ओके, विल मीट अगेन टुमारो।